Okay, great. So I'm Trey from UCSD and Christina from Sloan Kettering, and we also had this mandate to talk about predictive modeling, and our focus really to sort of distinguish our session from the others a little bit, but as you heard, not much, is on the modeling. And so really the question is, what does the PRS score model look like of 2025 or 2030 or whatever our goal is here? What do EQTL associations look like as of 2025 or 2030? One interesting note is that all these models, those and many, many more, are essentially these black boxes where you have G and maybe plus E, sometimes going in one end, P coming out the other end. That shouldn't be too surprising. But if you think about the importance of that statistical predictive model, that's basically what's in between right now. And so to convert G to P, that's going through necessarily every time some informatics. So the question again is, what should that piece look like in the future or the near future, and then what data, importantly, do we need to get there? And so I'll let Christina take over for now.
Okay, great. We started by listing a set of 5 to 10-year proposals to respond to, and there was great discussion. I'm not listing the proposals here. I'm just sort of synthesizing, summarizing some of the discussion. There was an agreement that as many institutes get involved in the genomics of disease, NHGRI has the opportunity to lead with integrative data sets and also lead the computational analysis and the methodologies for understanding data. And that whatever large-scale data generation efforts we undertake, they should be thoughtful and purposeful, and we should think what would be most informative given current biological knowledge. So within the room, there was strong support for collecting deep molecular phenotypes, epigenomic, transcriptomic assays in biological systems for training predictive models of gene regulation. There was some disagreement whether traditional cell line assays are sufficient or whether we should be moving into more biologically relevant systems, organoids, directed differentiation of IPS lines, but there was certainly support for this kind of work. Also, several advocates for pushing transcription factor DNA interactions, mapping these, continue to map these, understanding more quantitatively doing dose response. At the other end, there was some discussion, certain people advocating for collection of molecular phenotypes in GWAS subjects. Okay, so like the supersized version of GTEC, and I guess also discussed in Breakout 5. So these are different kinds of projects that enable different kinds of modeling and can be both appropriate, but it's different computation. There's some intermediates between these. So one nice suggestion, what if you take your current polygenic risk scores, take subjects in the tails and collect cells and use those as models for, multiple genetic effects that make you at risk for obesity or not at risk, protected from obesity. A lot of support for non-genomic data, like leaving the comfort zone of next-generation sequencing and looking at image-based molecular phenotypes, spatial single-cell RNA analysis, and also for interpreting the genome in terms of modern notions of cell biology, okay, like membraneless organelles, right? So there's different notions of function once you understand there's more than transcriptional regulation, right? And then we had, as one of our proposals, looking at cell-cell interactions and developing experimental systems for communities of cells and computational modeling, and it was not discussed at all. So that, maybe that's a 10-year plan because we have no ideas of how to do it. Okay.
Yeah, and just to finish out the report, so as Christina said, there was some agreement and some discourse on what are the main data sets we need. And I'm not sure this particular body is going to come down to these cell types or these patient populations with these layers of data. level proposal anyway, but we thought there were some broader take-home messages that had pretty firm consensus over the workshop or the work breakout session. One was mechanistic interpretability. So as you build these models, right now they are absolutely black boxes. The PRS score, you can pull out the top variants that contribute to that, but beyond that, you really have no understanding. The model offers no understanding mechanistically of what's going on. That reflects a lot of the comments we heard yesterday, I think, and to some degree today. So there needs to be models of 2025, just pick that date randomly between 2020 and 2030, need to be interpretable. Okay, now, as Jonathan, I think it was, pointed out, how interpretable and at what granularity depends entirely on the problem, and we don't want to script that here, but simply you want to be able to check the interpretable checkbox. Okay, point two, transferability across context. Huge discussion on the need for transferable models, and different people had different suggestions about what transferability means and what would be most useful. So we've already heard a lot of discussion about transferability across model organisms into humans, and ultimately imagine how great it would be if you had a PRS model that actually operated cross cell lines, cross evolutionary contexts, cross developmental time points and stages of aging. Of course, this is dreaming, but that's the point. That's the goal, is transferability across contexts. The other thing I like about that whole discussion that went on is it reflects an undercurrent of machine learning. Transferability is a big topic in mainstream machine learning at the moment. That gets me to point three, which is predictive performance. Another very large topic in the mainstream computer science field, of course, as all of us have undoubtedly heard, are the advances in deep learning. And Anshul, I think it was, pointed out sort of there's this tremendous opportunity to graduate from, say, the linear models used by PRS scores and other type approaches like EQTLs to more advanced statistical models, deep neural networks, and many other frameworks that are now blooming in the mainstream computer science field. And then finally, it was discussed, and it was a great point, that although we would like to have fully accessible data, and that's of course a huge requirement of building any effective model, is you've got to have accessible data, we also need to acknowledge as a community that that is going to partially happen, partially not happen over time. And one potential way you can advance this field is by advancing our ability to compute and use data even in their encrypted or partially encrypted form. So with that, I'll end. Anything from either Christina or the breakout session participants that has been missed?